Okay, very good. So let's get started. I hope everybody followed the links that I posted on Slack. So first I would like to show you the setup that we want to use optimally today. So on the one hand, on the right, you see you will have my stream open on your phone. Optimally, you would have it on full screen, and depending on your internet connection, if you're connected to your home Wi-Fi, which I hope you are, you should put your quality of the video as high as possible. Right? So I think the maximum would be 1080p, which is, I think, a very good quality, so you can see all the details in the presentation on your phone. Otherwise, it might be a little bit difficult. Okay. So if you're using your laptop, like you see in the presentation, you should have, for instance, the whiteboard open. Right. So let's see, let's go over to the whiteboard and let's check who's there. So if you've joined the whiteboard, I would like you to choose a color and write your name on it, anywhere where there's space. Nobody's done it so far? Okay, Kate, very good. I see Alex, Joshua, very good. Oh, okay. Everybody's a little bit more north. Okay, very good. Excellent. So I hope we can keep a little bit of order on the whiteboard. So if you so during the presentation I will have a couple of questions and some things to work with. So if you could keep your color, that would be great. You can choose to either write your name with your trackpad on your laptop, or you can also choose to, for instance, write with the normal text with a keyboard. Doesn't matter. Right, both is fine. Okay, so that looks good. The other thing, let's go actually back to the presentation. So the other thing that you will have open is, of course, Zoom for when you have a question, right? So probably the best time to ask questions will be when we have the whiteboard open, because people will be writing and there will be a little bit downtime and I won't be talking. So if you have a question, it's probably good to ask it then and just unmute yourself on Zoom, and I can basically hear you in my headphones and then answer on the stream. There, something that you have to note is that basically my reaction is a little bit delayed. The delay is around, I would say, 6-7 seconds. So for instance, if you write something on the whiteboard or if you ask something on Zoom, it will take a little bit for me to answer. So it's not like a real call or something. Okay, very good. I hope everything's clear. So let's see. For the other people, I think not everybody has the option or wants to, but for those of you who want, I shared the program PyMol. And I hope everybody has it installed and everything is working fine and you've downloaded the files that I shared in the Google Drive. If you have, maybe we can check it so I kind of know who is working along. If you could just type in the Zoom chat, right? If you're using it or not, that would be great. Right, so I'm going to wait a little bit. Just type yes or no. Okay. I see, okay. So some of you have not downloaded yet. I will give you a little bit of time. And in the beginning, I wanted to give you a little bit of introduction to PyMol because it's not as intuitive. I mean, PyMol is a super useful tool. Researchers around the world are using it to basically build proteins, analyze proteins, and do research. And today I wanted to just show you a little bit how you can use it. It has super many functions. It's a huge toolbox, which you can also, if you know programming, you could also basically write your own scripts and things like that. But today we're only going to use it for visualization and looking at proteins. And for that, it's not that difficult, but the user interface is a little bit difficult, so you have to get used to it. Let's see. So let's switch over in the stream to PyMol. Okay. So from the 3 files that I hope you downloaded, there will be one that's called Protein Workshop. So you will need all the 3 files that are in the folder on the drive. And if you open the Protein Workshop, what you see in PyMol should look something like what you see in the stream. All right. Again, let's do a roll call. Who can see the same as I do in their PyMol in the open window? If you see the same, type yes. If no, then type no. Okay. Let's see. Yes, no. Yes, I downloaded it without license. Does that work? The license doesn't matter if, as I wrote in Slack. So if you don't have a license, you can still use the basic functionality, even have a 30-day trial. But I mean, even afterwards, you can use the basic functions, just like viewing proteins and like manipulating them in the way that we're going to do today. For a little bit advanced functions for researchers, you would have to get a license at some point, but not for today. So all the dialogue with the license, you can just skip. It probably also, every time you open it, it will actually ask you for activation. You can just always click skip. It doesn't matter. Okay. All Right, I know Joshua had some problems. Is that fixed? Okay. Maybe? He hasn't replied yet. Theoretically, it should be possible with the iPad, I think. The input will be a little bit difficult because it kind of requires to have a trackpad or mouse. And if you want to input some text, you also need a keyboard, of course. Okay. So let's go through some very basic things. Okay, Gaku, yeah. The the file is called proteinworkshop.pse. Okay, so let's go through some very basic functions. So what you see just in front of you are basically three areas. One is for coding on the top, right? I told you before we can use Python to write our own scripts if you wanted to, if you want to learn coding, if you're interested in manipulating proteins and stuff like that. But for now, we don't need that. So there's actually a little bit of line between the main window and the coding window. We can actually shift that up a little bit so we don't need all the space for the coding area. On the right side, you will have basically, I would call that an identifier window. I don't know the technical term per se, but basically all the selections that you make in the protein, you can save them on the right. We will get into that maybe a little bit later. What's most important now, and for this workshop, is the main window, right? So this is your main way to interact with the protein that you've loaded into the program. So the very basic function is using your mouse. You can basically click and you can drag it, right? And basically, just look at it from different directions. Left, right, up, down, whatever you feel like. All right. Okay. Left click and then dragging around, right? The other one would be right click. Very self-explanatory. Up and down, in and out, like zooming. Right? So you can drag, move it around, and then zoom in and zoom out. Okay, so another one that's very useful is the middle mouse button, right? So right now you see it all basically turns around the center point, which is the gray line molecule which is there. But you can change that with the middle mouse button. You can basically just shift around left and right, up and down, and then the center point will be somewhere else. So it's not the molecule anymore. It's now within the protein. Okay. Sometimes if you look at proteins, it's very easy to get lost. So what I did for you in this file, I've saved different scenes that you can always go back to and look at. All right, so those are in the bottom left. I all gave them different names, and I will tell you to go to different scenes in part of the presentation, so we all can see the same thing, and I know that you don't get lost somewhere along the way. For instance, now let's just click on everything. Okay, so if you click on everything, you go to the saved scene with the saved settings, and it now looks quite different. It looks kind of pretty. It's like rainbow colors and a bunch of twisty lines, and yeah, still you can turn it around, you can go zoom in and out. That's all the same. Okay, so let's say you get lost. Okay, oh, now I'm here. I don't know like what to do. Just click back on everything. It zooms out and reverts to the old settings. Okay, I think that's, for now, that's what we're gonna need. Right, so if you're working along, just so you can, if you want, take a look at different angles and explore a little bit. Something to mention is also something that you see on the top. It's just a line of different letters. We will talk about that also later. Those letters represent basically the protein and its amino acids. Okay. If that is okay, then let's go back to the presentation. All right. So we will basically switch, or I will have to switch back and forth between PyMol, the presentation, and the whiteboard, right? For all of you to see and basically follow. See, so let's start with the whiteboard, right? So I give you the question because I kind of want to know what you already learned in school, what you know from maybe your parents or from friends. Just a very simple question: what are proteins, right? So let's go to the whiteboard. Yes, okay. Now we'll show you the little presentation on the bottom right. And I would like to know, basically now I will write just the word protein on the whiteboard. So what I would like you to do is draw a mind map around that. Basically answering, everybody can write whatever they want, whatever they know, right? And write down, what do you think a protein is, right? For instance, maybe somebody's into sports, they know, hey, protein, that's like whey or something, right? That's what everybody drinks to get muscles, right? Muscles. So go over to the whiteboard and I will wait for you to write down some answers. Okay. Okay, somebody's writing. Oh, okay. Chain of amino acids, very good. Try to keep what you're writing close to what I wrote on the whiteboard. Okay, very good. A molecular machine, chain of amino acids, macromolecules. Still waiting. There's so much you probably heard about protein. I bet. Organic compounds, okay. All right. Let's see. That's already very good. some of these use words that maybe not all of you are 100% sure what they mean, I guess, but it's good to associate them with proteins. So I'm not sure everybody in the workshop today knows what a macromolecule is. Amino acids probably, I mean, you find it very often on products, in foods, but maybe also the chemical basis is not that clear, I guess. Okay. Material of muscles, very good. Pavel, of course, knows what we're talking about. A molecular machine. We'll go into details of that maybe later on, not today. Carbons, very good. All right. Okay. Very good. So let's go back to the presentation. Okay, very good. So if I had to tell you in a very simple way what proteins are, I would say they're super long molecules, and they mainly consist of, well, it's an organic compound, so mainly carbon, hydrogen, oxygen, nitrogen, and sulfur. They have a very complex and a very sensitive structure. Maybe those of you who joined the summer program last year, they know that, right? That's basically one of the takeaway messages. And they're also able to make reactions happen faster. Right. So what that means, probably won't get to today, but sometime in the future. Okay, let's see. So in order to really understand what proteins are, we have to go a little bit into the very basics of chemistry. Right? So I want to make sure that all of you really know what we're dealing with and why what I'm showing you is how it is. Right? So, another question for the whiteboard. Right? In what ways can two atoms interact? Right? So on the whiteboard, let's see. What I'm going to do is, on the top, I'm just going to write two atoms, A and B. Okay, so how can A and B interact? So I want you to either draw it or write it down if you know anything. Okay, I say something on the bottom right. Maybe pull it a little bit closer to the atoms themselves. Okay, very good. Very good drawing. Okay, good. Any other ideas? Covalent bonding is a good point. That's the very basic interaction that we know that is very, very important for all molecules. Let's see, what else? Kipple charged salt. Okay. Very good. Yes. Covalent bond means sharing electrons, right? All right. So if we Think about that. Let's put a little bit of structure into all the answers when we go back to the presentation. So something that I saw very often is called a covalent bond, right? So in the pictures that you see in the presentation, you have, for instance, two types of covalent bonds, right? And you also have an ionic bond, which was what somebody else wrote, what you also call a salt. Right? So covalent bonds is basically when two atoms share electrons with each other, right? So if they're the same atoms, it's called nonpolar, right? Because they all share them equally, like in the left case, right? Oops, let's see, let's go back. So the atom on the left shares equally with the atom on the right, which is probably the same atom. If we look at the right picture, what we will see that they don't share them equally, right? So one of the atoms that share has basically more electrons around it. So like a bigger bubble, as you would say, or a bigger cloud of electrons. This usually happens when the atoms are different, right? Because atoms have a property called electronegativity. So if every atom is different in its electronegativity value. So if you put them together and they share electrons, it's probably not going to look very non-polar. It's going to look polar, right? So uneven. If they don't directly, well, this is a little bit the extreme case, the ionic bond. They also shared electrons, but the difference in the electronegativity is so strong that actually one gives the electron to the other one, right? So you have two atoms that are still associated because one is positive and the other one is negative, so they kind of attract each other. But one, the negative one, has the electron from the other one. Right? This is usually what you call a salt. Okay. So here's another example of an association or an interaction that can happen between atoms, right? That I haven't heard of or that I didn't see on the whiteboard. This is what you call hydrogen bonding, right? So if you think about it, hydrogen, I hope most of you know, is H2O, right? O and H, they're not equal when it comes to electronegativity. So the O basically takes most of the electrons around it and has a bigger electron cloud. What that does, it gives it a negative charge because it has more electrons, which are negative, and a positive charge where the protons are, or the hydrogens. So what that does actually in water, it gives it a special structure because positive attracts negative and so on. So the hydrogen is attracted to the oxygen and to another hydrogen and another oxygen and so on. So it makes a very complex structure, and it has basically the bond between the oxygens with the hydrogen are almost shared, right? So they almost equal distance between the polar covalent bond and the hydrogen bond, right? So usually, or sometimes even this one can just jump over to the other oxygen for a little while, and then it jumps back again. That's how much they share, because the difference of electronegativity is very, very high, and they can get very close, right? Okay, very good. So now I'm going to show you another interaction of not only atoms, but also like a little bit more complex molecules, right? So molecules can have different properties when they react with another molecule which is a little bit different, right? Maybe some of you have known, maybe some of you have not heard, but there's something called hydrophobic and hydrophilic. Hydrophobic means, like it says in the brackets below, I don't know if it's really readable, it means scared of water. So the green box is basically hydrophobic, and the blue ball is water. So when they come in contact, opposite to what you know, the drop wouldn't spread out. It would keep its ball shape because it's of course attracted to itself, like we saw in the slide before. Right. On the other hand, in the yellow box, if it's attracted to water, the water droplet will spread out as much as possible. Right? Because hydrophilic means it loves water, so it wants to have as much contact with the water as it can. Right? So if you, for instance, think about it, if you put a drop of oil, which is hydrophobic, into water, what it does, it doesn't spread out, right? What it does, it forms just a little circle. So to have as little contact with the water as it can. Okay. So, and below are two examples for that, right? One is, for instance, motor oil or cooking oil, or usually oils in general. And something that mixes very well in water, as we know, is, for instance, sugar, glucose, right? As shown on the bottom right. So we will see later those kind of interactions, right? Bonds, hydrogen bonds, hydrophobicity, like positive and negative interaction, are very important in proteins. So in the beginning, I just wanted to make sure you kind of understand what all of these terms mean. So, getting down to business. The basic part of a protein is an amino acid, right? So, well, why is it called an amino acid? Probably most of you have had different levels of education in chemistry. Probably most of you have not really gone into organic chemistry much, but actually the name amino acid makes a lot of sense. Because on the left, this formation of a nitrogen with two hydrogens is called an amino group, right? So the left side is called an amino group. The center is a carbon, and on the right side, this formation of a C with two O's and an H is called a carboxylic acid, right? So if you mix amino group and carboxylic acid together, what you end up with is the word amino acid, right? So very logical. Okay, to show you an example in the file that we have, let's go to PyMol. So in PyMol, I have basically selected one single amino acid for you. Right? And all you have to do is click the AS symbol on the bottom left of the black screen. What that will do is, like, basically an amino acid is going to fly into the center of the screen. Okay. So this one looks a little bit different than maybe what I showed you in the presentation, because now it's not 2D on on a screen, and it's not basically in written with the letters, but now you have different colors, and each atom is a ball, right? Like the model would show us like it would exist in nature, I would say. All right. So again, I would encourage you, turn it around, look at it from all kinds of angles, and just play with it a little bit. Zoom in, zoom out. I think it looks kind of interesting. Something that's not in this picture, you will see, are the hydrogens. However, what you see, let's turn it around, or if you get lost and you want to go back to the basic view of the molecule, just click on the AS again. So blue, in this case, is nitrogen. So this would be the amino group. On the right side, what you have is, here it only shows one O because the other one is connected to another molecule. So this would be the carboxylic acid. So, but there's something else, right? Maybe in the presentation you were wondering, what was the R, right? Maybe the R you wouldn't find on a periodic table, because R stands for residue, right? So maybe you have heard before, amino acids are basically the basic structures of a protein, but there are many different types of them, right? So the basic thing about them is the amino group and the carboxylic acid group, but there are different variations of them, and they all differ in this additionally bound atom, or whatever residue is attached to it. Right. So let's go back to the presentation. Okay, so we're talking basically here about this R. So, and for humans, there are actually 20, 21, depends on if you want to have like a very special one included. I would just say here are 20 different amino acids. Right? The R from the last slide is basically highlighted in this little box, or in these boxes. Right? So for instance, sometimes it's only an H. It's a very simple amino acid, right? Called glycine. I'm not 100% sure about the pronunciation. Or you have a C attached to it. You have 3 C's attached to it. You have sometimes a C and a sulfur attached to it. Sometimes it even can make a little loop, like proline with a amino group. Or some of them are actually quite long, like arginine or histidine, and so on. And as I told you before, since, well, if the atoms are all the same, you would call it non-polar, right? So here we have the big group of non-polar amino acids, because most of them, like the electron cloud is distributed very evenly. So not a lot of charge is produced. Not produced, but is present that would cause any form of polar attraction. Right? So probably if you were to take those amino acids, you put them into water, they wouldn't like the water very much. In comparison, you also have polar amino acids, right? And most of them have in common that they carry an O, right? So an O is a very polar group, so it makes them in total polar. Some of them also have a charge, so they completely take up a proton and get a positive charge, or they give away a proton and they have a negative charge, right? Out of curiosity, if we go back to PyMol, which amino acid are we seeing, right? So, and I would like you to answer that on the whiteboard. What amino acid do you see in PyMol right now? Okay, here it is. Which the chart? Okay, sure. I mean, it's maybe a little bit too small. Oh, I see. Okay, you mean the presentation. Okay. 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 Let's see. I'm gonna put a rectangle down so you can write your answers in there. If you look at the presentation, look at the different amino acids, and you look at PyMol, what amino acid do you see? Anybody? Nobody? If there are questions, now's the time to ask, right? Because we're on the whiteboard and people have time to write something down. And I'm also listening to Zoom. Ah, okay. Okay, some people maybe don't see the box. If you don't see the box on the right of the whiteboard, you have a hand icon. If you click that, you can drag the canvas around up and down and move and see different parts of the canvas. Okay, either glutamine or asparagine. Very good. Alanine? Alanine? Question mark? Very good. All right. So let's look at the presentation. So what we saw in PyMol is the basic amino acid plus the gray. The gray ball represents a carbon, right? So it's only one gray ball. So this is the extra that's attached to it, right? So the C. So the correct answer is alanine. Okay, good job to whoever found it out. Let's see. Okay. So, but maybe you've heard, I talked about it. Proteins are not only one amino acid, right? So they're very long chains. These chains are formed when the carboxylic acid group is combined with the amino group, right? It's actually an exothermal reaction that will lead a connection to, you would split off water, and this forms what you call a peptide bond, right? So as soon as you combine two amino acids together, the name of the whole molecule changes, and you call it basically a peptide, right? Two amino acids equal peptide. And everything that's bigger is also peptide, right? I don't remember who it was, but somebody once showed me, oh, I found a very, very long molecule with a very, very long name, right? So, for instance, for this molecule here, this would also have a very long name, because you would have the name of one peptide, one amino acid connected to the other one. And if you want to name the whole peptide, you have to just put them together. So proteins can be very, very large. So the names for them chemically would also be very, very large. But that's only on the side note. Okay. Let's see. Okay, before we go to this question, let's go to PyMol and let's look at it. Because I've also made a selection of that, and that's number 2 in the list on the bottom left. If you click on it, it says peptide. Right. So again, I encourage you to move it around, look at it from whatever angle you want. And what you see is I've highlighted each amino acid in a different color, right? So in this case, carbon is not always gray, but carbon is, for the left one, gray, in the middle one it's green, and on the right one it's pink. Right. So those are not 2 but 3 amino acids connected together to form a peptide. Right. What you also see here is the nitrogen and the oxygen, which forms the peptide bond. Okay, very good. Again, I would like to ask the question, what is the amino acid sequence of this peptide? Right? 3 amino acids. We found the first one out before, which is alanine. What are the 2 other ones? Again, I would like you to write your answer. Actually, let me go back. Yeah, write your answer on the whiteboard. Okay, let me make space. Use the same box as before. Right. Okay, what is the sequence of the peptide, or what amino acids are connected to each other in this, in PyMol? Okay, so because it's peptide, I would like to have 3 answers, right? The first one is definitely alanine. Second one, and then the third one. Typically, how you would write it is, for instance, alanine, then a dash, then the second amino acid, and then the third one. Okay, very good. Okay, there's one answer. Can we get another one? Okay. Let's see. Okay, let's go to PyMol again. Let's look at what we see again in the program, right? So this was alanine, right? The first one, the gray. Because this one ball right here is a carbon representing CH3. So this is an alanine. Here we have 1, 2, 3 carbons, a nitrogen, and an oxygen. Right? So if we go to the presentation, which one would fit? Okay, found it. So here is the basis, right? And then we have 1, 2, 3 carbons, oxygen, nitrogen. So, wait, sorry, you don't see my pointer. Okay, so here, the correct answer is glutamine. The second was glutamine, right? 1, 2, 3 C's, an O, and an N, or nitrogen and oxygen. Right? So correct is first alanine, second glutamine. So the third one, looks very similar than the first one, doesn't it? Right? So you have an oxygen, like the amino acid backbone, and the extra one is a carbon. So this is also alanine. So the correct answer would be alanine, glutamine, alanine for this peptide. So for instance, maybe let me check. I also have on the whiteboard, if you go to the bottom, you see a chemical formula from the very beginning. Maybe you have to zoom in a little bit to make it visible. I am going to put let's see. I'm going to put a thick border around it. Right. Maybe this one's easier. Right. So if you go to the presentation, this is a longer peptide. This is not only 3, but those are actually 1, 2, 3, 4, 5, 6, 7, 8. 8 amino acids. Okay. So challenge. Who can find out what this amino acid sequence is? So again, go to the whiteboard, and then I'll be waiting a little bit for the answers. Also, if you have any questions, type in the Zoom chat. If you don't want to talk, right? I'm also looking at the Zoom chat meanwhile. So anything pops up, I will also try to answer it. right? To make it more clear for everybody. Somebody? Okay, if we go to the whiteboard, let's just start with the first one. This is the first one, right? Okay, so the first one has, let me draw. 1, 2, 3, 3 C atoms and 1 nitrogen. Right? If we go to the presentation, which amino acid has 3 carbons and a nitrogen? Oops, sorry, I made a mistake. 4 actually. 1, 2, 3, 4. So 4 carbons and 1 nitrogen. Glutamine? We had glutamine before, right? Glutamine was 1, 2, 3, oxygen, nitrogen. It looks like the acid group and the nitrogen are attached to the same group. So you say in the chart it looks like they are attached to 2 different carbons. I think with the chart you mean the presentation, right? So the part this is referring to is this one, right? The nitrogen and the oxygen is attached to the third C in the line. Okay. All right. Same you can see in the PyMol model. Right? It's just upside down. Because you have to be a little bit good at imagining everything in 3D. Right? So maybe that is what makes it a little bit difficult. Right? So I haven't talked about it yet, but of course, if you look at the paper, you can make everything on the same side, right? And you can make everything look pretty and organized. If you look at it in 3D, it's actually somewhat flexible, and what can happen, the amino acids can actually flip and turn however they want. They can rotate between each other, right? Which very often happens, right? Just from like normal molecular movement. And also when they're fixed in a structure, then they're probably fixed in one of the orientations, like either up or down. but I'm not going to go into too much detail about that. Okay, let's look one more time at the whiteboard. I saw Jun already answered in the chat. I see somebody's writing very, very small. Okay, so if you look at the whiteboard, June is actually correct. This is the lysine. Oops. Right. So correctly, you would write lysine. Okay. Very good. So it's probably going to take too long to go through everything. I just wanted to kind of see if you guys understand. If it's difficult, that's fine. If you want to know more, we can go into more detail, for instance, in the mentoring. For now, we have to a little bit move on, or we won't get anywhere. Okay. So one more time. What we had on the whiteboard is a lysine, because you have 1, 2, 3, 4 carbons and 1 nitrogen. Right? Very good. So, again, another question. And I think this has nothing to do with what I taught you today. This is something that I hope most of you have learned in school, right? Another question or another thing I would like you to answer on the whiteboard. So the question is, how are proteins made? Right? I showed you before the slide where you saw, well, one amino acid is connected to the next one. If you split off a water, and it forms a peptide bond. But in nature, how does that actually work? How are peptides made? Okay. Again, let's use a box on the top. So I'm going to erase what's in it. And please write your answer in the box. No idea. Okay. It's okay. If you write no idea, that's good. I want you to interact. I want to know what you know. And if you have no idea, I need to talk about it. And if you don't write anything, I won't know if you don't know. Oh, very good. DNA, very good. Anything else? Okay. Ribosomes, very good. Okay. Pretty good. DNA can definitely be made, and is made in every cell every time. All the time. tRNA, very good. Excellent. And it's true, it's also a chemical reaction. Right? So the same reaction that I showed you on the slide is actually happening also in cells, right? To make peptides. So let's look. I mean, it got a little bit small, but this is on the whiteboard, this is the figure that I wanted to use to show you a little bit how proteins are actually made in a way. So on the top, you can just use the words that were in the box, because I think all of them you have already said so far. What's the thing on the top? I think most of you have seen that, and just write that in the left box, somebody. Okay, very good. I see it very, very small. Good. Excellent. That's DNA. Yes. By the way, there is no plural form of DNA. So it's not DNAs. It's just DNA. Okay, very good. Second one was already also found, right? So the difference between the top one, the DNA, and the middle one is, instead of T in green, you have a U in green. Uracil, found in mRNA, right? And this whole process of DNA turning into mRNA is called transcription. I'm just going to write it in here. Transcription. Very good. So then you have mRNA, right? And the mRNA then turns into, on the bottom left box, what is that? What is that? Very good. Yes. All right. So protein is a good answer. Maybe you could also say this is a peptide, right? So just a chain of amino acids. So actually, the whole process of turning mRNA into peptides is called translation. Right, just on the side. I hope you've heard it at some point. Very, very cool and very important process in every cell. Okay, what I haven't told you so far, actually, the peptide that you see here, it says met-il-ser, right? What is that? So every amino acid has a long name, but it also has a short name, right? Either it's a 3-letter abbreviation or only a single letter abbreviation. Going back to the presentation, 1 second. You can actually find them in this list that I showed you below, right? 3-letter and single letter abbreviation. So if it says met or M, it actually means methionine. Methionine. I'm still not 100% sure about the pronunciation. Right? The other one was Sir. Sir is, or S would be serine. Right. Okay, it's covered now, but the bottom left one. Serine. Right. Okay. So in a way, actually, the only reason we have DNA is to save the information on how to make proteins, right? Because the DNA has the equivalent code of a peptide chain encoded on it, saved on it in the nucleus. And the total of the DNA is basically all the information about all the proteins that are in one cell. Because proteins are basically what cells are in total, right? And how life works. So to keep this information and to be able to produce proteins and live, you have DNA, mRNA, and protein. In detail, there are many different things. It's a very complicated process. Perhaps somewhere in the future we can get a little bit into the thick of it if anybody's interested. Okay, going back to the presentation. So we answered how a protein is made. Somebody wrote ribosome, which is very good. That is something you can see here, right? So ribosome is basically a protein that makes other proteins. And it uses tRNA, which somebody else mentioned. And tRNA is basically like a carrier that holds one amino acid on the back of it, and it has another part of it which has a codon. And this codon then binds to the corresponding spot in the mRNA. And on the top of it, the chemical reaction happens, which then combines this amino acid with the already existing peptide chain, right? And as long as the mRNA is, as long as the reaction will continue to make a complete polypeptide chain. Okay, very good. All right, so okay, this is the same that we saw on the whiteboard. This is a polypeptide chain. Okay. Another question, a very simple one. I think everybody can answer. Okay, again, please use the box on the top to answer the question. I'm going to erase everything inside. In what environment do proteins normally live in, right? In the cells, in your body. What environment do they usually exist in? What do you think? If you don't know, write I don't know. That's fine. I don't know. Okay, that's fine. Anybody else know or not know? What I mean is, proteins don't exist just by themselves, right? They have an environment that they're in. And they basically have a surrounding, like we have a surrounding. I live in my home, and I live... If I would basically ask the same question about me, I live in air, right? Without air, I couldn't live. Okay, very good. There's a good answer. Also very good, right? So cytoplasm is a very good answer. Also, other proteins is a very good answer. What's cytoplasm, by the way? What's cytoplasm made of? There we have a very good answer. Okay. Excellent. So, correct answer is, right, if we go back to the presentation, proteins live in water, which is a polar solvent, which we talked about before, right? It's polar and doesn't like fat and other things. And there are also salts in the water, right? So like salt that you know from the kitchen, like sodium chloride, calcium chloride, other types of salts, phosphates, and other things. And also, of course, other proteins. Right? So all the things that affect a peptide are written here, right? So water, salts, and other proteins. So right after that, another question. What do you think would happen if we put a peptide with only hydrophobic amino acids into water? I just see extra crowded conditions. Actually, that's also true. You always imagine that the inside of a cell is very spacey and you have lots of distance between things, but actually cells are super, super crowded. Right? There's so many things inside them that there's not enough space to actually spread them out. So to answer the question that I just asked is okay, I'm waiting for you, Yukiko. Well, as we talked about, or as I showed you before, it depends on the amino acid composition what property the peptide has. Right? So on the whiteboard right now, on the bottom right, all I will do is basically draw you a representation of a peptide, right? Just a black line. So I'm asking of you, what would happen to this peptide if you put it into water? As an answer, I would like you to actually draw the peptide and how it would change, right? So if you think it's going to be like this, draw it like this, right? If you think it's going to be like this, draw it like this, right? Again, I will give you some time. Solved. What does that mean? You mean you think it dissolves in water? Is that what you mean? Okay, very good. Well, the peptide bond won't be broken by water, so it would not dissociate. That's something I can tell you. Okay, give you a little bit more time. Okay, excellent, excellent. Very, very good examples. Correct answers are I draw a circle around them, right? Oh, okay. There's another one, another correct one. Okay. So what would happen is basically the same thing that I told you about with either the water drop on the hydrophobic surface, or if you think about oil in water, right? Since it's hydrophobic, the peptide chain would behave similar to a drop of oil. That means it wants as little surface area with the water as possible. So what it would do is, some of you correctly said, it would basically coil up and get clumped up into a very, very tight structure, right? Because it wants to stay away from the water as much as possible. Okay, very good. Actually, for those of you who said chopped up, that's also a very interesting thought, because you're thinking maybe like chemists, right? You're thinking if those were not optimal conditions, it actually could revert back to single amino acids. So basically, if you, for instance, were to put in if you were to put in an alkaline, or in a base, basically, you could actually revert the process of the formation of the peptide bond and make it split up again, right? But if we're thinking about just normal water, which has a neutral pH, nothing would happen, right? It wouldn't split just up by itself. Maybe over a long, long time, right? But not in an instant. Okay, clumped up is the correct answer. So now I would like to first, let me erase everything again. Let's say we have the same peptide, but this peptide is actually not completely not completely hydrophobic, right? But it would have, let's say, a hydrophilic end here, right? Okay. And let's say a hydrophilic part here, right? So let's say we put this peptide into water. What happens? Right? And again, I would like you to draw. Draw what happens to it. No, hydrophilic. Sorry, my mistake. The green part is hydrophilic, so likes water. Okay, actually very nice. Okay, so basically soap is an interesting statement. Okay. Let's see. So some very good ideas. Basically, let me give you the answer. What would happen is that's what it would look like. So in a way, actually, I would kind of count purple drawing, right? Because basically the hydrophobic part is separated from the hydrophilic part. It's also true in this drawing that water would surround the parts that are hydrophilic, right? So in the correct version, what you can see is the middle again tries to have as little surface area to the water as possible, and the hydrophilic parts, they want to have as much contact as possible, so they would basically stick out of the whole bundle, right? Okay, very good. Let's go back to the presentation. So this is one type of interaction that I just told you about, right? So an interaction that is happening between molecules or atoms also happens within the complete chain of the peptide, right? So in this case, what we just talked about is the orange box in the middle, right? So hydrophobic interactions or hydrophilic interactions. There are also other types of interactions which are very important for what this protein looks like in water. One of them is, for instance, an ionic bond. Right? We talked about before when I showed you the diagram of the different amino acids. Some are positive, some are negative, and they attract each other. So if you have a positive one on the one end and a negative one on the other, they would kind of find each other, and they would create like a circle. Right? There are also covalent bonds within proteins, right? So some amino acids have the property to associate with one another, right? And they can be combined. In this case, or it's cysteine. It's one specific one. And this also, we could go very much into detail in how actually those are linked, because there are also specific other proteins that can do that. And also something that's very, very important within the peptide are hydrogen bonds. So not only, so basically they share this proton. Those two oxygens share this proton with each other. They have a stronger connection than the rest. So all taken together, this makes the formation and the the arrangement of a peptide very, very, very complex. All right. And this is the part where I would like you to go to PyMol again. Because we have a peptide that we can look at, and we can see what this looks like in real life. Basically. So in PyMol, I would like you to click on unstructured. So what you're going to see is we will have a zoomed out view of the arrangement of a peptide, right? So it's not a line in this case. This representation is basically the atoms and their angles between them, and a little bit more complex. Actually, let me show you. You can also choose a line representation. Oh, not this one. The ribbon one, yes. Okay, that's what it would look like if we were to look at it like we did on the whiteboard. If you want to have the same view on the right side, you have to click on unstructured, and it will select all the amino acids that belong to the part that you see. And you can click on H on the right panel next to unstructured, which then gives you a pop-down menu, and you want to hide everything. So H stands for hide, and you can choose what you want to hide. In this case would be everything. So then you just have the selection with the pink dots. And then you can click on S next to it, which stands for show. And in this case would be ribbon. Show ribbon is this type of representation. Right. If you're done, you can also, again, like before, look around. Right. So instead on the whiteboard, we only can, of course, look at 2D. This program, you can look at what a peptide would arrange itself in 3D. All right. So this peptide, which if you're interested, you can actually find out the amino acid sequence in the top. Because I told you in the beginning, there's this little line of just letters in a row. Letters represent the amino acids. And if you have highlighted the unstructured in the right panel, it will show you in the top part which amino acids those are. So in this case, in this protein, this peptide with these amino acids arranges in this way, right? In 3D. So something, it's not straight, has a little bend, and another little bend, and so on. So let's look at what we just talked about in this case. So if you're lost or you misclicked something, again, just click on unstructured, everything's back to good, like to the scene that I want you to see. So in this case, let's think a little about why does it look like this? All right. With all the information that I've given you so far, let's find out. So, let's see. What do we have sticking out here at the end, right? So if we start from this end, what amino acids do we have, right? So we see that this part, this whole peptide chain, has a lot of red, right? Red means oxygen equals polar equals hydrophilic. So just as in the example that I showed you on the whiteboard, this part does not clump up right? So it's free. It's just floating and is probably surrounded by water, right? So a lot of O's, and so on. Let's go to a different part. Let's see. So we have a fold in here. So what would make this peptide fold in itself, right? For instance, if you say we have a mainly hydrophilic chain, actually the hydrophobic amino acids would fold onto themselves, would connect, would seek each other. In this case, one of the main non-polar amino acids is this one. It's called phenylalanine, right? And this one is actually very close, right, to another one, which is this one on the bottom. Those are both non-polar amino acids, right? So this one on the bottom is an alanine, like we saw from before, and actually close to another alanine, which we had in the beginning, which is represented still with the balls and the sticks, right? So those like each other, so they will search each other out and want to be in proximity. Okay. So again, click on the scene. And something, well, you might be saying, okay, well, this one over here, the green one, that one's not non-polar. So why is it so close to all these other ones, right? Why does it like support this fold in a way and is like in this pocket? The reason for this, you can see if you click on the fourth scene in the file, which is unstructured H bonds, right? So in this program, you can also visualize hydrogen bonds. In this case, they are the yellow dotted lines. So with the yellow dotted lines, you see the reason that the green amino acid is so close to the other ones is that it can share protons. It can share the hydrogens with these other atoms, right? And basically, sharing atoms means a tight bond, and if it basically can bind very tight, it also has a energy preference. I mean, we will get into that later, perhaps. But the reason is, but what I want you to see is hydrogen bonds are very good, and if a peptide can form hydrogen bonds, it will definitely do so, right? In this case, it does so by making this curve, right? Plus, it also supports the non-polar amino acids that are connected, right? Something else that you can see, here's another bend, right? Between the last amino acids. This is something a little bit special. Here's an amino acid called proline, which I showed you before. Proline is a little bit special in that it has this ring, right? So, okay, this one also has a ring, but not attached to the backbone. So the backbone is basically what you saw as the line. The residue, well, there are rings as residues, but not within the backbone. I told you before, the amino acids, they can flip. They can basically move freely. So, but proline is actually not as free in its movement because it has this ring. So proline basically usually is the beginning of a turn in the peptide, right? So now with everything I told you, you can basically understand why does this peptide look as it is, right? So with all the things we discussed today, the basic chemistry, we talked about amino acids, their sequence, their properties. And now in a real example of a protein, a real life protein, you can see this is, all these rules apply, and you can make sense of something that probably looked pretty senseless in the beginning, right? So this structure has actually a deeper meaning behind it. And in a way, there are also many uses to go from the properties that each amino or peptides have to actually predicting how they will fold, right, in real life. But that's also something for maybe another workshop. Okay. The green part is hydrophobic, you said. No. If you, just to answer the question, if you think about the categorization of amino acids that I showed you, the green one was glutamine, right? So glutamine is a polar, or a polar amino acid. I mean, of course, in this case, a long chain of carbons is kind of nonpolar, but because it has the oxygen and the nitrogen on the end. Oh, sorry. Okay, yeah, it's polar. All right. Okay. I think this is a good time to stop for today, because otherwise I think it would get a little bit too much. And I would also like you to remember today what we talked about today, because we will actually need that kind of information if we want to go into the next part of this workshop, right? Looking at the bigger structures, right? We started from the atoms, we went to amino acids, now we're at peptides, and we will go into bigger and bigger structures. But still, to understand those, we need the basics, right? That we talked about today. Okay, so with this, I would like to go into an question answer part, right? Or discussion, I would say. So if you have any questions, let's go to the whiteboard and either type them in Zoom, ask him in Zoom, or maybe also write them on the whiteboard. We will see. You can choose what is the best way for you to ask the question. Okay. Anybody? Any questions? Any concerns? Anything that's unclear? Anything that you would like to know more about? Now's the time. Okay, so the question is, are there more forces that govern the configuration besides hydrogen bonds and hydrophobicity? Very good question. Yes, there are. So what I told you about are the major ones, I would say. Maybe from chemistry you would remember some of them, right? So they have to do with like van der Waals interactions and like dipole interactions, things like those. Basically, all interactions that affect how atoms interact also affect a long molecule of atoms, right? And how it forms structures. Right? So all the things you know from basic chemistry are also important when you think about bigger molecules and how they fold. I hope that answers the question. Actually, also, let me take a look at the YouTube chat. Other questions, anyone? Can we make proteins with lab equipment? Very good question. Yes, we can, right? There's a whole field, I would say, in biochemistry that focuses on protein synthesis in the lab compared to what happens in real life. Of course, we can use, for instance, if we insert a DNA strand into an organism, as I showed you before, this would turn into RNA, and this RNA would turn into a protein. So basically, we've made a protein with DNA, right? And we made it in a lab. You can also make it with chemical reactions, right? It's been a long time, so I don't remember that much about it anymore, but yes, it definitely exists, and it is a very active and very, very interesting research, which produces a lot of results which see application in daily life. Right? For instance, you could also, for instance, produce insulin, right? So a protein that diabetics use to deal with their disease and to regulate their blood sugar. Can we design our own protein structure and make it? Yes, right? So for instance, the program that we use is not only for viewing, it's also for editing, right? So if you wanted, you could just make your own protein structure in the software. If it exists like this in real life is another question, right? So there's also a whole field that basically focuses on predicting the structure of proteins from a theoretical amino acid sequence. Yes, and those would be proteins that we've never seen before. So you could take just a sequence that you think of and that you think is unique, as it doesn't exist in any other form in nature or in other proteins, and you can fold it and you can see what kind of structure it makes, and yeah, you would have a completely new protein. Another question from Zoom: do peptides fold as they are made, or is the full peptide made before it folds? Okay, very good. That's something we will come to in the next workshop. So that's also the reason why I showed you a little bit how a ribosome works, because, well, peptides are assembled one by one. So as the chain starts to elongate and is released from the ribosome in its natural environment, it starts folding step by step, right? So it's not that the whole peptide is made and then folds all of a sudden. It's a process, right? As the chain gets longer from the ribosome, it starts folding and assuming different structures and until it then has its final form. Very good. Other questions? Okay. I think if there are no other questions, maybe not now, but I would also like to get some feedback about how the workshop today was, how you like the stream setup, the maybe the whiteboard, the exercises, the connection with Zoom. I'm looking forward to a lot of feedback to make it as as pleasant as possible and as interactive as possible, so you can learn a lot better. Okay, so I hope I will see you guys next time. Bye bye.